Good morning, everyone, and thank you for the generous introduction. It's truly an honor to be here in this amazing department and meeting with all of you. I had a great discussion last night with few faculty, and it was really great gathering. And before we dive into today's topic, I just wanted to show that this is a snapshot of University of Minnesota. And although there is a rumor that we are always covered with snow or with freezing temperature and we need snowmobile to get around, all of them true. But we also have sparkling weather like this. And actually Minnesota is ranked third in terms of outdoor activities in all the states in the US. I'm seriously very excited to be here today to talk about some of the most important development in the perioperative pain management and their possible impact on clinical practice. I do not have any financial or non-financial relationship to disclose that are related to this presentation. And here you will see some familiar sightseeing landmark. You see the Taj Mahal, you see Statue of Liberty, Eiffel Tower, and Burj Khalifa. This sightseeing landmark has no relationship with the content of this talk. They just represent that 4 key articles we are going to discuss today that could be a landmark article in our field and possibly impact on the clinical practice. So, sorry, although it seems to be a grand round of your next vacation, but it's not. So, all right, this is a picture that you all recognize. This is Taj Mahal. It's located in India and built by Emperor Shah Jahan in 17th century. It took 20 years, 20,000 people, and construction budget that I guess will make all of us sweat. And story goes that Taj Mahal was built by Emperor Shah Jahan as a great symbol of love for his wife Mumtaz. And the name Taj Mahal came from Mumtaz Mahal to Taj Mahal. It sounds very sweet, right? But people who are not a big fan of Shah Jahan, they say that there's no love here. He just wanted to glorify himself because he did have multiple wives. Whatever the real story is, Taj Mahal stands as one of the greatest landmark in history. And today, under this grand backdrop of the iconic symbol, we'll talk about a landmark trial in the regional anesthesia that has shaken our field by lawsuit, stock market crash, and a lot of other drama. So there's no love story in my talk, just cold and hard evidence. So, and this study is perineural liposomal bupivacaine is not superior to non-liposomal bupivacaine for peripheral nerve analgesia, published by, first authored by Dr. Nasir Hussain and in Anesthesiology Journal in 2021. Liposomal bupivacaine, as you know, is very different from non-liposomal bupivacaine. It is designed to deliver controlled levels of bupivacaine and is composed of naturally occurring biocompatible lipids. It encapsulates the bupivacaine molecule in a suspension of multivesicular liposomes and releases bupivacaine over time and achieves targeted analgesia at the target site and provides safe and consistent level of bupivacaine. So up to this point, this is a company insert in the vial, and company is right. There is no argument. There's nothing wrong with that statement. But company also claim that this liposomal bupivacaine eliminates the need for catheters and pumps that may hinder recovery. It also claims that it extends analgesic duration while reducing the need for opioid. And they did not provide much evidence for these two claims and that require further investigation. So this is the study by Nasir Hussain, published in Anesthesiology, as I mentioned. So in this study, by the way, Dr. Hussain, when he published this study, he was a CA3 resident, which I think quite impressive, because a CA3 resident did a meta-analysis and published in a journal, Anesthesiology, as a first author. We don't see this very often. Whatever it is, the study authors did systematic review and meta-analysis of the 9 randomized trial, and they compared liposomal bupivacaine with the non-liposomal bupivacaine for peripheral nerve blockade as not field blockade like TAB blocks or plain blockade. So primary outcome of their study is the difference in area under the curve of the pooled 24 to 72 hour resting pain severity scores. What they found is 24 to 72 hour area under the curve pain score is statistically but not clinically significant. That means there is a small difference which is statistically significant but not clinically meaningful. So here the story goes more interesting. When they removed one trial that is industry sponsored, they find no difference in pain severity score at any individual time points, opioid consumption, time to the first analgesic, or patient satisfaction. So now you might ask, what is the reason of removing a trial? What is the problem with the industry sponsorship? And why they need to cherry pick the articles? And this I will unpack in the next slide. So industry sponsorship introduces significant bias how the study was reported, conducted, and written, published. It has been shown in multiple trials. It has been shown that industry-funded trials likely to favor the sponsor's product with a relative risk of 1.27. And Turner and his colleagues showed that publication rate of the positive data, that means positive when intervention or drug worked, 98%. When this does not happen, that means intervention or drug they used did not show any efficacy, only 37% of the studies are reported. And out of the 37% studies, only 8% accurately represent that studies are actually negative. Rest of them say that either they may be underpowered, maybe some technical glitches, maybe statistical sample size was too low. They say everything except admitting that this study simply did not work, or the intervention or drug did not work. And we have seen the similar trend in liposomal bupivacaine study. When liposomal bupivacaine was found to be superior to comparator group in 46% of the study that are conflicted, that means industry sponsored. But it was found to be superior in only 11% of the non-conflicted trials. So it is pretty clear that whoever funds the study can have a major influence on study results. So after publication of the Nasir Hussain study, which we discussed, and another study by Brian Ilfeld, who had identical conclusion, and both of them published in Anesthesiology, Pacira, which is the maker of liposomal bupivacaine, sued American Society of Anesthesiology and asked for retraction of the articles. And their argument is trade libel. Trade libel is a legal term which means intentional disparagement of the quality of the services or products of the plaintiff's business or damages to the plaintiff. So let's step back for a second. The first author, Nasir Hussain, was a CA3 resident at the time of the publication. And the guy who even did not enter into academic clinical practice, why he would have gut or like brave enough to sue a pharmaceutical giant? It doesn't make any sense. And district court ruled that drug maker failed to state a claim for trade libel. Pacira appealed the decision and lost again. The story did not end here. In October 2023, ASA announced the receipt of 2.5 million donation from Pacira to advance education and innovation to its members. So hopefully with this donation, conflict between Pacira and ASA will be resolved, because as you know that sometimes money talks. But sometimes science does not only change clinical practice, it also makes tremor on the stock market. Here you see that the stock graph of Pacira in the last few years, and you see that I marked some of the important point here. Here the Nasir Hussain and Brian Ilfeld study published, which showed the meta-analysis showed no difference in the liposomal bupivacaine and non-liposomal bupivacaine. And here Pacira sued the ASA and district court ruling in October 2025, February 22, and after that you see the stock started to slide, and it sank by 65% of the original value over the next few years. So it's pretty clear that good science and good evidence can have a ripple effect that is far beyond the clinical practice. So what is the main takeaway from all of this Taj Mahal study? So we see from these two trials, one of them I did not discuss because of the short of time, the liposomal bupivacaine did not show clinical superiority over non-liposomal bupivacaine for peripheral nerve block. But does this surprising to you? How many think it is surprising? When Pacira submitted IND for approval of their drug, they actually did not need to show that it is superior than the competitor drug. That's not a requirement. They only need to show that it is better than sham, which means saline. And since liposomal bupivacaine contain local anesthetic, it will definitely be superior than sham, that means saline. That's pretty low bar to clear a drug. An FDA advisory panel raised this concern. They voted by 6-4 against the expansion of the indication of liposomal bupivacaine. Due to this reason we discussed, that lack of comparator group, lack of safety studies, and lack of evidence for opioid sparing that company was claiming. So part of the reason, but FDA concern was, advisory panel's concern was overruled. Part of the reason is we are in a opioid epidemic, and there's a tremendous pressure to clear any drug that has potential to reduce the opioid reliance. So in other words, we are offered to have a luxury car which doesn't drive any faster than a Toyota Corolla. And the cost difference is pretty significant. The cost of 266 milligram of liposomal bupivacaine is $334, whereas in equal amount of bupivacaine only $3. So now we are in Paris with the iconic Eiffel Tower, which is a symbol of elegance, connectivity, and love. Completed in 1889, it was originally criticized by some as become the eyesore. But over time, it became a symbol of masterpiece of engineering. Today, it stands out just as a symbol of France, but of connection, creativity, and of course, of romance. Just as the Eiffel Tower stands out, reshaped the Paris skyline, and technique I am going to discuss now can reshape our skyline of the regional anesthesia. And let's talk about a fascinating study by Brian Ilfeld, published also in Anesthesiology, percutaneous peripheral nerve stimulation for postoperative pain, a randomized, sham-controlled pilot study. So before I dive into this paper, I want to tell you that peripheral nerve stimulation is not a new concept. It has been in the practice for a very long time. What is exciting and new is its application in acute postoperative pain management. And this was possible due to miniature design of the PNS device. And as you see in this picture, so placement of this PNS is very simple and similar to the single shot nerve block. Under ultrasound guidance, you use a twin needle very close to the nerve. Then using Sildinger technique, you place the electrode very close to the nerve and remove the needle. And other end of the lead, either you can attach to the small pulse generator, or in some model, you can bury under the skin and then stimulate wirelessly. So now see how well it works. Let's look at the evidence. As mentioned, this is the study by Brian Ilfeld, and in anesthesiology, a randomized multicenter prospective sham-controlled trial after ambulatory orthopedic surgery. What authors did, they placed the electrode into the nerves So pre-operatively, the team placed an electrical electrode to target a specific nerve depending on the surgical site. Sciatic nerve for the foot or ankle surgery, and femoral nerve is ACL repair, and brachial plexus for rotator cuff repair. And then after the surgery, before the surgery, they gave single shot local anesthetic, and same as our nerve block. But after the surgery, they stimulate the one group with electrical stimulation and another group with the sham stimulation. What they find is the median opioid consumption in the neuromodulation group is only 5 milligram, whereas in the sham group is 48 milligram, which means a tenfold decrease of the opioid consumption. And in the next slide, this opioid consumption is extrapolated over time, and as you see that red bar is red line is a neuromodulation group and blue line is the sham group. As you see here, neuromodulation group has a significantly less opioid consumption over time, and it is sustained for 7 days. So in summary, the neuromodulation PNS is a very promising neuro technique for postoperative pain management, but we need more research. So far, all the results we get from only one team. But something we need to consider that this technique has several advantages over traditional pain management methods. First of all, you don't use any local anesthetic, so there is no chance of motor blockade. There's no chance of local anesthetic systemic toxicity. There's no medication bag required, so there's no catheter management, which is a significant cost to the practice. And long-term use, you can leave the leads up to 60 days in some models, which is FDA-approved, but catheter-based analgesia, which we normally give for less than 7 days. So now we are in the Dubai, in front of the Burj Khalifa, which is standing tall as world largest, tallest building. It is not just an architectural marvel, but it's a symbol of ambition, innovation, and frankly, a healthy competition. Because what I am hearing that some other countries like Saudi Arabia is now planning to make some towers which are taller than this building. But height aside, this Burj Khalifa is a architectural marvel. It integrates residential, commercial, and entertainment spaces, a true multifunctional structure. And under the backdrop of this towering achievement, we are going to talk about some towering technique in our own practice, like thoracic epidural analgesia. In my view, it's the thoracic epidural is a Burj Khalifa of regional anesthesia, specifically for abdominal and thoracic post-surgical pain. And you look at the two important articles published in the British Journal of Anesthesia that triggered some friendly debate. And now let's read this article titled, Therapeutic Efficacy of Intravenous Lidocaine Infusion Compared to Thoracic Epidural Analgesia in Major Abdominal Surgery, a non-inferiority randomized control trial. And the author concluded that IV lidocaine infusion is non-inferior to thoracic epidural analgesia. How many of you believe this study? How many would think that it could be possible? Can you raise your hand? Exactly, no one. So, same thing. After publication of this article, there is a storm in the social media that how journal, like prestigious journal, like British Journal of Anesthesia, published this article. Because it doesn't really align with our own clinical experience. So what happened here? How these two asymmetric, which is anesthesia technique, properly placed thoracic epidural analgesia and IV lidocaine infusion shown statistically similar. One explanation is, which me and my colleagues proposed in our commentary to the British Journal of Anesthesia, which I'll tell you in the next slide, that a lot of epidurals are not placed without imaging, may not be actually in the right place. So if you combine a failed epidural or suboptimally placed epidural with those that placed correctly, you essentially diluting the efficacy of the technique. And then average could make it is possible and make it similar to the effect of IV lidocaine infusion. It's possible. And let's take a look at what we wrote regarding the article. In our commentary, we stated that there is a high rate of failure of thoracic epidural analgesia, and we cited multiple studies that shows that thoracic epidural failure can be as high as 50%, which is staggering considering how central this technique in our practice. And the epidural failure is believable if you look at this picture. Extreme choroid angulation of this thoracic spinous process make it extremely difficult, challenging to navigate the twin needle into the epidural space. And sometimes when you push, it can be slipped to the paravertebral place. And other thing is ligamentum flavum are not fused in some of the thoracic epidural, upper thoracic epidural place, which can make high chance of intrathecal entry of the epidural needle. So when you interpret the trials, thoracic epidural to other modalities, you have to factor the real-world variability and failure because it absolutely affects the outcome. So now obvious question is how can we improve this thoracic epidural failure? And Kumar and colleagues provided a great summary of the some practical techniques that can help improve the success rate. One of them going to discuss which we incorporate in our practice is the contrast epidurography. It is a very simple process. You place the epidural like similar way, like you do here in the block room or preoperatively, and after the surgery, when patient in the postoperative recovery unit, you inject contrast, and we take a picture, and based on the image analysis, if we think it is not in the right place, we pull it out and place it again. Sometimes we place second time or third time, but we make sure that after patient leaves postoperative anesthesia care unit, patient has a functioning epidural. So ideal is, of course, this technique does come with a learning curve because reading contrast epidurography is not something you do as a regional anesthesiologist. And here you need your chronic pain colleagues like Dr. Nathalie Strand, I'm not sure she is here in the audience, and your other chronic pain colleagues. That can be a huge help. It's definitely best to place this epidural under fluoroscopic guidance. And I know that some practices already incorporated it, but it depends on your case volume, and you need access to the C-arm and workflow logistics. Other techniques like epidural waveform analysis and electrophysiological stimulation looks promising, but they are still early or experimental stages, and we need more data before bringing them into routine practice. So bottom line is thoracic epidural analgesia is a valuable technique for abdominal and thoracic surgeries, but it is associated with a significant failure rate. Some studies claim that it can be up to 50%. So techniques such as electrophysiological stimulation, waveform monitoring, and X-ray epidurography can significantly enhance the success rate. So now you see the iconic Statue of Liberty standing tall in the upper New York Bay. Originally, it was conceived as a symbol of deep friendship between France and the United States, and a celebration of the liberty, specifically abolition of the slavery after American Civil War. Over time, it became much more. It became a beacon of hope for millions of immigrants who came to this country for a better life. So under the backdrop of this iconic great symbol of freedom, I want to talk about an FDA-approved non-opioid analgesics that could give us some freedom from the opioid reliance for in postoperative pain. Let's dive into it. So the story for development of this drug traced back in Lahore, Pakistan, and clues came from a firewalker family. A group of researchers from United Kingdom are visiting Lahore, Pakistan, and they noticed that something pretty remarkable. They found that a street boy was walking on burning coal and also stabbing his body with knives, and he does not show any sign of flinching, distress, or pain, nothing. Naturally, this caught their attention. So multiple further investigations led to the groundbreaking discovery, which was done at the Cambridge Institute of Medical Research, and the result published in Nature journal. The research found that the boy and several of his relatives had a genetic mutation, and this mutation inactivates the NAV 1.8 gene, which was critical for sensing pain. Now, that discovery opened up a brand new target for pain relief and set the drug developer a very challenging journey for 2 decades. After 2 decades of journey, the BX548 developed by the Vertex Pharmaceuticals, and which was approved by the FDA, and this VX548 Suzetrigine blocks NAV 1.8 sodium channel, which exclusively expressed in the primary afferent pain sensing neuron. And by the way, we do have a lot of sodium channel blocker, but they are non-specific. So they have a wide range of side effect and doesn't work very well. But NAV 1.8 exclusively expressed in the primary afferent neuron. So unlike opioid, which acts broadly on the central nervous system and cause a wide range of side effects, VX548 or Suzetrigine works right at the target, at the source of the pain. That means it has the potential for effective pain relief with fewer systemic side effects. Let's take a look at the real world data. So this study published in the New England Journal of Medicine, and it is a double-blind, placebo-controlled, randomized controlled trial. And it's a major milestone for this drug. And in this study, patients undergoing abdominoplasty received either VX548, 100 milligram loading dose, then 50 milligram twice a day for 48 hours. And results showed that drug provided significant postoperative pain relief up to 48 hours. And this is pretty exciting result. Not just because the drug worked, but it worked without typical opioid-related side effect. BX548 or Suzetrigine showed almost identical results in patient undergoing bunionectomy. This consistency across the different procedure is really promising addition to our multimodal pain management toolbox. So in summary, FDA approval of non-opioid Suzetrigine or VX548 for adults with moderate to severe acute pain is a crucial milestone for acute pain management, but we need to look at post-marketing studies to better understand its long-term effect, tolerability, and safety profile. Thank you for your kind attention. I'm going to stop here.